My name is Ida, and I am a sophomore in college. So it's been a while since I've participated in any sort of science Olympiad event, but um, uh, today I hope to just like share a little bit about, um, I guess, how to. Actually, you know what? Before we start talking about the event, how many people have done an ID event before? Again, just like a little raise of hands if you've done an ID event. Okay, so it seems like the vast majority of you haven't done an ID event, which is okay. So entomology hasn't been in circulation since I think 2015. So because of that, I wouldn't expect anyone to have done it specifically, but events like ornithology, forestry, fossils, and rocks and minerals are like basically the exact same thing, just with like slightly different. Sorry, people are coming in and out. I'm like, this should be okay. It's basically going to be the same exact format. So when you're preparing, you can just keep that in mind. I'm going to share screen. How do I do that? Okay, so you should be able to see my screen now. And you should only be able to see this one tab. Okay, so yeah, this is entomology for division C. Okay. So, okay, yeah, so this is like the original index that Femke, our lovely organizer, has made, but I changed it around slightly. So first for the rules sheet, anyone who has done an ID event before knows that you have access to a 3-read binder, which basically you can bring any kind of information that you want. In addition, you get a field guide, and also for some specimens, you're allowed to use a magnifying glass. And I know that in some particular tournaments, they're going to provide you with a microscope. So it would be good to know how to use a microscope. In ID events like these, you're basically going to be presented with a lot of different specimens. And these either could be actually in real stations, they're actually going to give you bugs, or it's just going to be like a presentation with bugs. And you're going to have to identify it by class, order, family, and common name. And then additionally, after you identify them, you're going to answer like a lot of questions about the bug. So they're going to ask you maybe about where it lives, or they're going to ask you specific things about like its morphology, things like that. So as you can probably tell, it's very rote memorization based. The more time you spend literally just going through flashcards and memorizing what they look like and facts about them, the better off you will be. So there's not a lot of, I would say there's not a lot of like it's not very skill based. So important things to think about. Since this isn't really skill based, there aren't very difficult topics. I think they're just some things that we might not think about generally. And the first thing is how important it is to organize your binder in a way that makes sense for you. So for people that have done ID events before, you know that you have to compile a very big binder of information. Quick show of hands, how many people have made a binder for an ID event before? I know some people just slay. Okay, so a lot of people know how to do this. I think the wiki also says this too, but from my years of doing like ID events, I think there are like 3 main ways people choose to make cheat sheets or their binders. And the first way is to have both images and information. You tend to like front load. So if I was going to make like a specific sheet, like maybe for like a grasshopper, for example, um, the first page of it would just be like various angles and various pictures of my genus. And then the second pages, I would talk about the habitat. I would talk about the different kinds of like each feature. And I would talk about the specific facts that you need to know about it. You could also just have images. So you would memorize most of the facts. And then the images, you would have more images so that you can probably identify more with more specificity. And the last thing is just having information. So that means you would be very familiar with what each of the insects looked like, and you would use most of your space to specifically, you have more space to have facts and things like that. What I found are especially useful when making a binder are making charts that map differences and similarities between each of the species. Because I feel like when you look at a lot of these questions asking you things like, this particular species has like how many sets of wings, or what kind of like mouth does this kind of species have. Sometimes it's hard to like go through all of your notes and look for that specific information. But if you have a specific chart just dedicated to the different kinds of mouths or the different kinds of wing shapes and things like that, then it's a lot easier to find. And also I think it provides you like some sort of intuition when you're memorizing things. So that's something to keep in mind when you organize a binder. And a second thing that I think might be overlooked in this event, just like looking through it, is the human impact part, because I think it seems to focus a lot on anatomy and environment and things like that. But from what I can tell, or from what people have said in the past, is that a significant portion of the test could also go to, human impact. Are you asking specifically about human impact? Because I can take a look for you. Mm. Entomology B. Yeah. So it seems that human impact, and like how insects interact with human life in general will be covered on both exams. Yeah. So some things to keep in mind for human impact are looking at positive impacts such as pollination, how they can be food in some areas, how they can control populations of harmful insects. And then on the negative side, they can be vectors for many different kinds of diseases. They can damage crops and buildings, like termites can gnaw away at wood and things like that. So what I've seen is that it's important to maybe make a chart for the types of diseases that different insects can carry, and also yeah, I think that would be an important thing. And also medicinal uses and things like that. Okay. I think the biggest um, part of preparing for ID events after you have constructed your binder is just practicing with it. Yeah, I think that might be the most important aspect, because after you have compiled all of your information, like when you're in a setting where everyone has a binder and everyone has probably spent months preparing it, the thing that differentiates you will be how fast you can recall that information and how well you can use your binder and your field guide, right? Like everyone has a field guide. You basically have like an infinite wealth of knowledge. So I think it would be really good to practice with how you find things and how you source things. Since you have two people, maybe one person memorizes a certain number of orders and families, and then the other person takes the other half. Or one person memorizes specifically like IDs, and then another person remembers the facts or the key features of each of each species and things like that. So unfortunately, there are like hardly any practice tests out there for entomology, which makes sense because it hasn't been in rotation for 10 years. So I made 3 very commonly seen questions for you guys. So I think I can flash one on the screen and you can work through it. How many people have started studying for this event, actually? Great. Okay. Oh, basically everyone. Lovely. So you have knowledge of this event. Um, actually, you probably have more than me because when I did all of these, I did fossils, I did rocks and minerals, I did forestry, but I never had entomology. Um, so this is great. I wrote 3 questions, um, and I'll give you guys maybe like 3-4 minutes to work on each one. You can raise your hand when you're done, and then we can walk through each one. So the so if you want to get your notes out, this is a great time to practice using your notes. This is your first question. This is like the most common one. It's just going to be an ID question. Right now you're seeing something on the screen, so unfortunately you can only see one angle. But yeah, but usually you should, if you're in a setting that actually gives you the specimen, you'll be able to look around. Maybe you'll be able to use your magnifying glass. But yeah, have fun with this one. Raise your hand when you're done, and then we can talk about it. Okay, right now I'm seeing that 2 people have finished. So I'll give it a couple more minutes. Remember, when you're done, just put up your hand so I know when to continue. Um, actually, that might be. Let's give it 2 more minutes and then everyone write your answer in the chat. That'd be fun. Okay, I think we spent a good amount of time on it. Let's all drop our answers into the chat. and we'll do a little chatting, a little comparing. That's real. You don't have to know. It's all good. Okay. Anyone else have any answers? Okay. If not, so you were right. Slay. Okay. Yeah, y'all did great. It is a pygmy grasshopper. Um, so this particular one does live in marshes. However, I don't know, according to Wikipedia, most um most pygmy grasshoppers do reside. Like there's the most biodiversity in rainforests, which makes sense. So I think you would be safe if you just said wetlands. And yeah, and this particular one is a female all-shaped pygmy grasshopper. And I think that the from what I remember in like previous events and things like that, like fossils, it was good to just like look at like a shit ton of images and different angles of particular families and particular genuses. And that's how I kind of just built intuition for what things look like. But you can also remember things like technically, like I remember this, the information for this particular one saying that what differentiates this from like what was the other kind of grasshopper? But like the most common grasshopper is that like the front part of like the middle segment, like the thorax, is really long versus a common grasshopper tends to be a lot shorter. So you can remember things technically, or you can just look at a lot of different pictures and build your intuition from that. So this is an example of an ID for those people who haven't seen it before, and I would say that this tends to be the level of specificity that you should get to. Okay, the next type of question we have is a dichotomous key. Does everyone know what a dichotomous key is? Okay, so I'm glad y'all are being so honest because I know what it is. Oh, great. Awesome. We get to learn how to do one. So dichotomous keys are basically, you can think of it as like a little tree that helps you like whittle down in specificity in order to identify certain things. Um, so I can give you an example of a dichotomous key. This is a dichotomous key for leaves. So basically, you have like an order of operations to go through, and slowly you can narrow down what kind of leaf it is. For example, if I take a look at this leaf here, um, if I just take a look at A, and I go to step number 1, needle leaf, non-needle leaf. This is a non-needle leaf, so then I go to 3. Is it simple or compound? It's simple, so then I go to 4. Smooth or jagged? It's smooth, so I go to 5. Ah, okay, can y'all see the leaves now? Lovely. Okay, thanks for telling me. Yeah, so this is a dichotomous key for leaves. So I'm going to go through it again. If I take A, for example, it's not a needle leaf. So then I go from 1 and I jump to 3. Here gives me 2 options again. It's the die for the 2 part. And I see that it's a single leaf. So it tells me to go to 4. I go to 4. It's smooth instead of jagged. So I go to 5. Is it lobed or smooth? It's smooth. So you know it's a magnolia leaf. So that's how you do it for leaves. And for insects, it's going to be the same way. Um, and so if we're going to kind of reverse engineer this with like the things that they already have, you can take a look at the specimens that you've been given and start grouping them, right, by characteristics. So I see how this person started. Okay, well, I see one needle leaf. I see two needle leaves, and I see several smooth leaves. So that's how they started grouping them. And then they look, okay, well, some of these are compound and some of these are simple. And then they group them that way. So now you know what a dichotomous key is. I'm going to give you 5 bugs, and you're going to try to do the same thing. Um, from the very limited amount of tests I've seen, it seems that you have to ID these yourself for the most part. Um, so I'm going to give you guys some more time. First, try to ID everything, at least down to the family. Wait, let me just make sure. Yeah, try to ID everything down to the family. And then after you do that, start putting them in groups. See what belongs to certain groups, and then go through there. I'm gonna see if there's a way. Yeah, and this is like a simpler one because I specifically made it so that some belong to the same order, and some belong to, like, two of these aren't even like insects, so they should be easier to group. And for dichotomous keys, a good rule of thumb is that you're always going to have one less step than the amount of specimens that you're given. So for the 5 specimens, you should have 4 steps, and each step has like 2 choices. So I'm going to give you guys some time um, to do this. Let's say, maybe, yeah, just like raise your hand when you're done, or if you think you have an answer, and we will reconvene. This one will take a little longer. What do you need to find in it? I think for a dichotomous key, the important thing is to help someone identify it. So you basically, to construct one, you just need to find ways to group your specimens and then ways that they differ from other things. So like here, they were able to, I need to switch it again. So if we go back to the leaves again, you can see that you can, one defining characteristic of the needle ones. I think you should give um, I think you should give the characteristics that are specific to those taxonomies. Because I think the idea is to help people narrow down by characteristic. Mm-hmm. And while you guys work on it, I'm gonna go make sure too. So yeah, but first, if you don't even know how to, the first thing to do would be to ID these first. So before you even construct the key, take some time to do the ID. And while you guys do that, I'm going to Sorry, I forgot to switch it back. Alrighty, who here has done or at least taken a crack at it? Great. I'll give it one more minute and then we can talk about it. Actually, let's do this. Yeah. Right now, I'm wondering, can people see my pointer, like my clicker? Okay, great. So how about let's go through each one of these and first see if y'all ID'd them correctly. We can, again, basically practice IDing again. So what did you guys say for this one, the first one that my clicker is going around? Yeah, that's right. Lovely. Okay. What about the one underneath it here? Yep. Slay. And then this guy? Yep, I'd hope you know this one. And then this guy? Yep. And last one. Actually, for the katydid, what is the scientific name for the katydid? Just for practice. Oh, yep. Yep, that's right. Slay. Okay. Now let's take a look at the dichotomous key. If it looks slightly different, that's totally fine. But here are the ways that I found to differentiate them. Okay. So the first thing, the first thing is I was looking for like the broadest category. So one thing that kind of differentiated them was that some of these, oh my God, some of these were insects and then are like part of the same class. And then the other one was I can never pronounce these, class Entagnatha, but they're not true insects. So that was the first level of distinction that I made. And so I put that in the first like step. And then I would say like one of the big distinguishing characteristics is, do they have mouth parts that are kind of concealed within like the lobes of their head, or do they have exposed mouth parts? So if they have concealed mouth parts, then we go to step number 2. Oh, sorry. Yeah, if they have concealed mouth parts, then we go to step number 2, because then you have to distinguish between the oh my God, the proturans and the diplurans. So, and the main difference between the two are that, oh, did someone leave? Okay, I guess someone left. Are that the diplurans have well-developed multi-segmented antennae, and proturans have either very like atrophied antennae or no antennae at all. So you've reached like the end level and you've identified 2 specimens. Now on to the next, which is that the rest of these have exposed mouth parts. This one made kind of easy because 2 of these are part of like the grasshopper and cricket category, and this guy is a roach. So what separates between the roaches and the grasshoppers? Roaches have less well-developed back legs because they don't really jump, and the grasshoppers do. So that's your order of distinction here. And number 3, well-developed back legs, you go to 4 for further distinction. Smaller back legs, that's your cockroach, and you've identified that one. Lastly, in number 4, you just need to distinguish between this guy, your katydid, and your short-horned grasshopper. And the main difference is that the katydid has a very long antenna, and most short-horned grasshoppers have short antennae. So this is a very simple example of a dichotomous key, and I did some more looking, and yes, so the reason why we don't make the key based off of taxonomy is because you're using the characteristics, you're using the key to determine what the taxonomy is. So your actual key should use characteristics that specifically distinguish each taxonomic bracket. So I hope that that made sense. Does anyone have any questions on this particular problem? If not, we'll move to the last type of problem that I've seen. Okay, great. So the last problem is super straightforward. It's just a labeling problem. This is taken off of a wiki that I found, and I've just boxed out some of the body parts. I think for these, you don't necessarily have to memorize all of them, just memorize like the basic things like head, thorax, abdomen, and then like main parts of them. Like you should know where the eyes are, you should know certain parts of the mouth, but just have like a couple pages in your binder dedicated to diagrams of things, because they'll also ask questions about like specific insects. They'll be like, this part of the thorax is longer in this insect and is shorter in this insect. Which are the insects? So I think you should memorize it, but also don't fret. Keep lots of diagrams in your binder. So just take some time to identify this, and then I'll go to the next slide and you can check your answers. And then we'll do a wrap up, and then that will be most of it. Not necessarily. Some don't give word banks. I just gave one for this one because there were a lot of parts. But yeah, some won't give a word bank. Also, to highlight the importance of like just having a lot of different diagrams in your binder, like have as many as you can, because a lot of test makers um, like they're honestly just like college students, so they don't have a lot of time. So people recycle diagrams. So chances are, if you have enough, you're going to have the exact diagram in your binder. So yeah, like some of the tests might use this diagram. So they probably won't give you a word bank, or I mean, they might, but if you have the diagram in your binder, like it doesn't matter. Yeah. But also, because not everyone will give a is it all the bugs? As in, does this pertain to all of the bugs? Okay. I think this this is a diagram of insects. So all insects should have these parts, but they might look different. Yep. Okay, I'll give it one more minute and then I'll switch to the next page so you can check your work. All right, I'm going to switch to the next page. Just do a look through, see what you got right, see what you might have missed. Okay. So now we're going to do a little wrap up, and just as you go on forward, so quick show of hands, how many people have started assembling their binder already? Okay, so like a good amount of you. That's great because I mean, yeah, regionals are still in a while, right? But it's important to have your binder like as early as possible, no matter how rudimentary it is, just so that you can spend the rest of your time adding more information, going through practice tests, and then based off of those practice tests, add more information, and then also memorizing. I think it's important, especially I remember doing fossils, how literally just running through Quizlets like all the time was so helpful because when everyone, again, reiterating, when everyone has the same amount of information, how quickly and accurately you can ID things, because you need to ID them in order to answer like all the like the other questions. So how quickly and accurately you can ID things is probably like the most important thing. So go through Quizlets, put multiple images, like for example, for like I don't know, like the pygmy grasshopper, put like 4 different images of it like on different flashcards so that you're used to seeing like different types of pygmy grasshoppers from different angles so that you're not like you don't just have this one conception of a pygmy grasshopper in your brain, you know? So run through Quizlets over and over again till it's like second nature, and then IDing will be so easy for you. And then practice tests are also super important for ID events, but because there aren't that many, um, I would say like go get a field guide and like quiz your partner with the field guide. Or I know several people, if you search up entomology, science Olympia, or just entomology flashcards, Quizlet, people have made several sets. So, and also there is, if you go on the Science Olympiad wiki, for entomology, scroll to the very bottom, there are additional resources. And there's this one website, it's like bugs.com or something. It has like, but essentially it's just a place where you can go look at all different kinds of bugs. There, quiz each other with like images from there, watch videos. Yeah, but speed IDing is very important. So spend a lot of time just drilling it into your brain, and then you should be good. Okay, so here are some resources that I have compiled. So the entomology ID list is a great place to start if you haven't assembled a binder before. If you want to take a look. So there's no website for that, unfortunately, but if you literally just searched it up, if you just search it up, some will come up. There's no website for that. But yeah, for entomology, there's this lovely thing on SciOly.org. If you need help getting started on your binder, um, it sorts them by orders and families. And then, for example, if I click on this one, it literally has all the basic information for everything that you need to know. I think the only thing that it doesn't have is it doesn't have the order Entonathra one. They have all of Insecta, but they don't have the first couple. So you have to find those. But literally everything else, use this list. Copy paste it in, remember things. If you have to no, I don't think so. Because the rules are 2-inch or thinner binder. So if you have a 1-inch binder, they're gonna count it as your thing. And yeah, but you can fit as much as, and also you can't take anything out of the binder. So there's that. However, since you're allowed a field guide, you can annotate it as much as you want. So if like you want one person to hold on to the field guide and mainly use the annotations and another person use your binder, you could do that too. But yeah. So yeah, but you can go through this list and literally just use this as your starting point. It has all of the basic morphology and everything. It tells you about like how they grow, their specific characteristics, where they live. It's up to you to put in images and things like that, though. I would say that. Okay. Yeah, so that's very, very helpful. The wiki, some amazing bug person from years past has compiled every, I would say this is very the wiki for entomology is very valuable for getting started on like what facts do I memorize? Because I know there's like this big list of like you need to know ecology, you need to know about like anatomy, you need to know this person breaks it down pretty nicely for you. So that's a great starting point. Um, here are basically the only 3 practice tests that I could really find. And I didn't want to use practice test questions today because then you wouldn't get more practice. So go through these, but beyond this, I guess you'll have to look for Quizlets. If you can find any test sets from 2015. But yeah, they're kind of hard to find. How many questions are there? I could take a look. They vary a lot. I'm assuming that I'm gonna ask Femke if she can share it because I'm assuming y'all found this on like a mailing list, right? So I'm sure it can be shared to you guys. Um, let me take a look at how long how long these practice tests are. I remember for fossils though, we had 10 IDs and then around 2-3 questions per ID. So pretty fast paced for what, 15 minutes? Slay! Thank you, Femke. Okay, so one of the practice tests has 16 stations, and 3 to 4 questions per. Uh, so I get like the idea is that because everyone has a lot of information, they're testing like how they're finding like the highest score is how well you know your information and how well you know your binder. So get lots of practice, memorize as much as you can, be very strategic about your memorization. Um, and yeah, lastly, get a good field guide and annotate it. So this one linked is like the official one that like Science Olympiad endorses. But this is the Audubon Society one. But there are several other ones. For fossils, I use Smithsonian, and I think that one's thinner, but I enjoyed that one because most of my information was on my um, binder anyways, and I kind of just liked it for a little extra reference or any species that I might have missed. But yeah, field guides are important. Um, and yeah, that's it. Thank you so much for coming. I'm so glad so many people are interested in bugs. And good luck on all your events and all your invitationals, and have a good one.